Hello everybody, today we are going to learn the recombinant DNA technology from chapter 3. This recombinant DNA technology is a very important part in the history of biology actually. This is like a groundbreaking work, a breakthrough. So therefore you have to pay attention to this lecture, and it is actually very interesting chapter. So you can look forward to it. Alright, this RDT is the abbreviation of recombinant DNA technology. So from now on I'm going to call it as RDT instead of this full name. Okay? So this RDT was initiated over the lunch discussion. It's interestingly, Dr. Boyer from University of California in San Francisco and Dr. Cohen from Stanford University, they met at a conference and they had lunch together. Okay, that was the beginning of RDT. Isn't it interesting? At that time, Dr. Boyer had this technique. Let me explain it in more detail. He was able to insert any gene of interest, it's a foreign DNA, into a vector. You know vector, you already learned it. The vector from bacteria, okay? Actually, the vector can be obtained from not only bacteria, but also from other sources like yeast and insect cells, etc. But bacterial vector is the most easiest one to manipulate actually. So now let's say you have this foreign DNA and you open up this vector here with restriction enzyme. Okay, you will learn about it in the next slide. You can cut a vector with restriction enzyme like that, and through that open area from that cutting you can insert this foreign DNA. So this is a resultant recombinant plasmid. Okay. On the other hand, Dr. Cohen, in each term, he was able to insert a certain vector into a bacteria, which is called transformation. Okay, he had this technique. So, combining the techniques that they have, what can you do? You see here, Boyer can produce recombinant plasmid, okay? And Cohen can put this recombinant plasmid into a bacteria cell. So this is the product of their co-work. Isn't it interesting? This is the beginning of RDT. So why is it so important, this RDT? Why is it so important? Why? Because you can produce any gene that you want in the bacteria, which is a cheap source. You can grow bacteria and at a cheap price in a large quantity. Whereas if you were to grow some mammalian cell, it costs a lot. Mostly, for example, let's say if the foreign DNA is like insulin DNA. Okay? You know insulin, what insulin is? Cure for diabetes. Okay? So let's say you want to produce insulin a lot. Okay? So normally the insulin is the gene from eukaryotic cell, like mammalian cell. So would you want to produce the insulin from mammalian cell in a large quantity? it's gonna cost you a lot. Okay, but if you use this technology, you can produce insulin, for example, from E. coli in large quantity. Okay, so that's why this is a breakthrough work. So let me go further with the discussion as to why this RDT is so important. Okay, so before RDT, everything was so non-specific. Okay, they didn't have the tool, or they didn't know how to introduce certain gene into some cell, okay, and produce that in large quantity. No, they didn't know how to do it. So what did they do? They knew at the time certain mutants can produce some protein that we want. You know, not every mutant is paralyzed. Okay, some mutants can be superior to normal phenotypes. Okay, so that time before RDT, the only thing that people could do was to introduce some mutagens to create mutants. Okay, so that was quite like trial and error approach, and it was quite random. You don't know which mutant will produce which type of phenotype. You never know. So if you are lucky, if you get lucky, then you get some good phenotype from some mutant that you tried. And usually that doesn't happen quite often. So, you know, you get, most of the time you get inferior phenotypes. So, you know, it is tedious, time consuming, and costly. You have to try over and over again until you get what you want. That's what it was, okay? So, that time, without knowledge of genetic information, they just had to try many things just to improve the existing inherited property. That was the best thing that they could achieve. All right, so what happened after RDT? You know, now you are able to manipulate the gene, the target gene. It is highly specific. All right, so you can produce what you want out of the gene you have in large quantity. Okay, that's why it can create highly productive strains. And mostly they are like bacteria, and the most common bacteria is E. coli, as you know. Okay, and as I just explained, large scale production. All right, and it is multidisciplinary training because it requires not only microbiologists but also chemical engineers, just like you. Okay, why? Because it's a manufacturing. Manufacturing requires chemical engineers. All right, so there your role comes. All right, and after RDT, of course, a lot of new business came into being. Okay, you know, like lots of biotechnology companies were established, over like 8,000 worldwide. Why? Because, you know, they have certain gene that they want to produce for clinical purpose, like for example, inhibitors or antibodies or growth factors. You name it. There are many. There are many proteins that are applied for clinical purpose. Okay, let's take a look at the development of RDT. You know, to be able to do the genetic engineering work, you should first know the structure of DNA, of course. And as you all know, Watson and Crick, they discovered the structure of DNA, and from that important work, they won a Nobel Prize, as you know. Okay, so After that, Boyer and Cohen, they established the RDT, which we learned just now in the previous slide. And right after that, in 2 years, Kohler and Milstein, they were able to produce monoclonal antibodies. This is very important, which will be explained in chapter 6. Okay, you have to remember that. And Genentech, I think this company was established by these two guys, which I'm not so sure about it, but maybe you can search from the internet. The name is not that important anyways. They established the company, like biotechnology company. And the company produced human insulin. I explained this, right, just before, from E. coli. Okay? And there are many other issues here. You can look through this history. And one more breakthrough was here. That happened in 1988. PCR method was published. This one will be explained, like you will learn it in maybe next chapter. This is also very important. You have to keep that in mind. And the detailed human chromosome, the map of human chromosome was published. This is another groundbreaking work. And eukaryotic organisms' chromosome were sequenced. Okay, sequencing is to know the order of base of the DNA, like T, C, G, etc. You know that. This is the sequencing. That was possible. That happened in 1996. This is not that long ago. It's like quite recent. Okay. And then there are other important issues too. T cell is related to immune system, and you can look through that. Okay. And now that you learn RDT, you have to do something with it, right? You know, RDT is all about manipulating DNA. So to manipulate DNA, what do you have to do? What should you have? First of all, you should have DNA. Okay? Well, of course, these days you can buy the DNA, like isolated, purified DNA from some manufacturers. There are many manufacturers that are selling these DNAs. But let's say you don't know the sequence of the DNA, then what should you do? You know, nobody is selling that DNA. You know, nobody is selling the DNA that we don't know the sequence of, okay? So what should you do? You have to first purify, isolate and purify the DNA, and you should have the DNA before manipulating that, okay? So how do you get the DNA? From where do you get the DNA? Of course, from the cell. To be exact, from the nucleus, okay? You have the DNA here, right? So, well, if it's a prokaryote, then the DNAs are all exposed in the cytosol. But anyhow, you have to first break the cell to be able to obtain the DNA. So, well, if it's a case with bacteria, then you have to first figure out which kind of bacteria it is. As you already learned, as I explained in chapter 1, there are two types of bacteria, remember? Oh, you don't remember? Yeah. Bacteria can be classified into two types, depending on the structure of their cell wall or cell membrane, their surface in short form. Okay, so the one with peptidoglycan wall is called gram-positive. Remember? And the one without peptidoglycan structure is called gram-negative. Remember that, right? So this is about, let me change the color here. This is about gram-positive bacteria. First, you have to remove peptidoglycan cell wall using lysozyme. Lysozyme can digest this. Okay, then since the peptidoglycan layer was removed, the underneath cell membrane has to be broken up, right? So you can burst the cell membrane using detergent such as Triton X100. There are many others. Okay. Let me change. Let me erase this. It's a little bit messy here. All right. And okay. And if you have plant cells, then you first have to. Most of the plant cells have some thick cell wall structure. Okay, and they are very rigid and hard to break. So you have to shear that mechanically, you know. And after that, the inner cell content will be smeared out. Then there must be some membrane here too, so you have to digest the membrane with enzyme. It's the same thing here. And if it's animal tissues that you have, then the animal tissues don't have cell wall. It has only cell membrane on the surface. So you just add enzyme to digest, break the cell membrane. Okay? So now you have cellular liquid that contains DNA, RNA, and protein, and some fragments of cell wall and cell membrane, which is mostly lipids. Okay, so now you have to separate them. You cannot just carry this all along, just all together. No, you have to separate them. How? Of course, we have to do centrifugation. After centrifugation, you can separate them. And mostly these, let me change the color again here. This part, these ones go down. After centrifugation, they go down to the bottom. And these ones here comes up here as a liquid. It's called spinatant. Okay, so what do you need this the bottom one? That is called the cell debris. They are the garbage. Okay, this is garbage. You don't need it. You don't need this. Eh? What you want is this upper part. This is spinatant, which contains DNA, RNA, and protein. There are mostly they are important, right? And let me erase this whole thing again. It's so messy. Alright, so now you after centrifugation, you have this cell debris and this. Okay, change the color again. And this part here. And now what you want to do is to extract DNA alone. Okay, you want to remove these two other ingredients. So what should you do? The most easiest one first is to remove protein first. Protein. How? By extracting with phenol. If you add phenol here, then this one is separated into two phases. The phenol has got higher density than water, and this water contains DNA, RNA, and this phenol contains protein. Okay, so you have to do the centrifugation again, which is not written here, but you can easily guess it. Centrifuge. Then you can take this upper layer, which has DNA and RNA only. Okay, DNA, RNA. Then what can you do now? Now you have to remove RNA only. Then you can get purified DNA. How do you remove RNA? You have to digest it. How? By applying some enzyme that can digest it, right? The enzyme named RNase, the full name is ribonuclease, can digest RNA. Anything ending with any enzyme ending with ase in its name is the enzyme that can digest something written before. So if it's written, what is written here? RNase. It digests RNA, for example. Okay, let me give you another example. For example, protease. It ends with, let me change the color again. It ends with ase, right? And what is written before? It's written, you can guess it should be protein, right? So what does it digest? It digests protein. Okay, now do you get that? Okay, now let's go on to the discussion on restriction enzymes. You must have seen this before, right? In the previous slide when we are talking about RDT. These restriction enzymes are the key for the genetic engineering technology. Without them, you cannot manipulate the DNA. Why? Because they can cut the DNA. This is the DNA, and they can cut and reseal. All right, so these restriction enzymes can cut the DNA in these fashions, two fashions. Okay, so one is leaving this type of blunt ends. Blunt, literally, blunt means leaving a clean cut without any part sticking out. Okay, whereas these sticky ends, they leave these parts that are sticking out. Okay, so let me change the color here. These sticky ends, they tend to stick back to each other here. Why? Because they're complementary to each other. T likes to go stick back to A. Also, this T likes to stick back with A. Same goes for others too. Okay, so usually these sticky ends are more common than blunt ends, and also these sticky ends are more effective in recombining the DNA. Why? Because they easily stick back. Okay, but blunt ends, since they have this clean cut, since they leave this clean cut, it's not that easy to reseal these broken ends. It takes more energy. Okay, now let's check some of those enzymes and the recognition sequence or site. Okay, you don't have to remember the name of these, but some of them appear quite often, like for example EcoR1 and BAMH1. You will automatically just remember them. And this recognition sequence, for example, they have different sequence here. Each enzyme's got their own specific recognition sequence. They have all different recognition sequence, and the length of the sequence are all different, and okay, let's say these slash lines, they are the cutting site. These enzymes cut here, leaving these sticky ends. And for example, another thing that you have to know is this, like for example, N is non-specific base. It can be anything, G or C, T, A, whatever. It can be any of them. So it's highly non-specific, but the length should be same as this. For example, for BGL1 restriction enzyme, okay? So let's say now you have these two sticky ends from these restriction enzymes. After they cut, you have these sticky ends, and they tend to seal again. When you anneal them, they tend to reseal like that. Okay. But you have to here, you have to see that not everything is fully recovered. Here you see still some broken up part. Why? Because you know these are the backbone here with a phosphodiester bond. How does it look like? Let me draw like that. You see some base here and something here, and this is phosphodiester bond, and another base down here. It's getting a little messy, and here the same thing repeating again here. Okay, let me remove this. It shouldn't be here. All right, so let me change the color here too. And this here, this is broken here. This is broken. This shows what this is. Okay. So now you have to recover this phosphodiester bond here again, and here this OH correspond to this, and this P is this with minus here. Okay. Plus, I'm sorry. So what happened here? Like you have to recover this bond by applying this ATP. There should be ATP to give energy, and the enzyme that is doing this job is called ligase. This is ligase. Ligase recover this phosphodiester bond. Okay, so you can complete this recombination job, DNA recombination job with DNA ligase. So first you have to cut with what? Restriction enzyme. Let's say you have this factor and this, sorry, let me change the color here. This 14 DNA you cut, these are cut fragment, and you have to seal again with the ligase. 2 steps, okay? So now that you have some DNA fragments after applying restriction enzymes, so let's say you have this DNA pool here, and you added restriction enzymes, then you will get a bunch of DNA fragments because they did the job of cutting the DNA, okay? Some are longer than the others, some are shorter, and you will have a DNA pool with various lengths, okay? So now you want to separate them, okay? You want to separate them. Then what would you do? What would be the principle of separating them? Of course, simply you can say that like they have different size, different lengths. They have different size, okay? So you want to separate them depending on their size, okay? How would you do that? For that, here comes the electrophoresis technique. This is a technique. This illustrates the electrophoresis technique. You will, right after that, you will watch a video that explains the protocol, experimental protocol of this technique. But for now, let me explain to you the principle behind this technique. Okay, you need, first of all, you need this electric field. You have to apply electric field, and this is the minus electrode and this plus electrode. And as I just mentioned before here, this DNA, let's say, well, this figure is not so good. I drew it, but anyways, this DNA has got this whole length with the phosphodiester bond, a series of phosphodiester bond, and each phosphodiester bond has got this minus charge. So overall, DNA is charged with what? It is charged with minus. Okay, DNA's got minus charge. You have to remember that. All right, here, all these DNA fragments are all charged with minus. Okay, so this should, if you put this DNA sample here with lots of DNA fragments here, okay, then what would they do if we apply the electric field here? Of course, they will, since they have minus charge here, they will migrate from minus electrode toward plus. This is a common sense, right? All right. So if it's for example, if the fragment is longer, then would you move fast? Okay, let's say we have two. Let me remove these things here. Let's say we have these two, sorry, with different color. Okay, we have these two different fragments with two different lengths. Then this one will move faster than this one, right? This one will move more slowly than the short one. So eventually, after completing this protocol, then the short one will go down to the bottom, and the longer one will be in the upper part. So let's say, well, these standard DNA fragments are available commercially. You can buy that. You can buy this sample from any manufacturer. Okay, and they have this standard band. These samples got these bands here to indicate which location is for a DNA band of which size. All right. So for example, let's say this is 2000 kilobase. What do you mean by kilobase? Kilobase. What do you mean by kilobase? Kilobase. Base is base. You know one base. For example, like G or C or guanine or cytosine or like thymine, adenine. These are all base, right? So kilobase means this unit's got like 1000 base there. So 2000 kilobase means 2000 multiplied by 10 to the 3 bases. All right. It's not clear here. Okay, so overall, how many zeros here? 6 zeros, yeah? So 2 million bases. It's quite long, all right? And this must be 10,000 billion base. So let's say now, let me erase it. It's a little bit messy. Let's say now you have this DNA sample, and you see this A sample's got 2 bands, after running this electrophoresis, you obtained two bands that one of each corresponds to almost 10,000 kilobase, and the other one corresponds to almost like 6,000. Okay, so you have these two. That means this A samples got, you have two different fragments. One is 10,000, the other one 6,000. And now the other sample B, you don't know what kind of DNA are there, and you don't know their size either. So after this electrophoresis, you can know after that. So you obtained three bands. One of them corresponds to almost like 8,000. The other one almost corresponds to, should be like 3,500. Looks like that. And this one is a little below than 2,000. Let me check. So should be about like 1,500 maybe. Yeah. From that you can learn your samples got which size of DNA fragments you can learn after all this experiment. And before going on to the video, let me briefly explain the little modification to this electrophoresis. One is PFGE. Let me change the color. This is not visible. PFGE, which is pulsed field gel electrophoresis. This is good for separating large pieces of DNA, because these large ones here, okay, let me change the color here. Let's say you have only large fragments, then they will stay upper part here. They will not go down here. You will see only these large ones overlapping each other. Then you want to separate them, okay? For example, like they are more like larger than 10,000 kilobase, most of them, and those are the samples you have, right? Then what should you do? They apply pulse. Okay, here. The pulsed field means like it's not like a continuous field. You apply field and pause, and then apply it again. You give pulsed field. Then this band go when the field is applied, and then pause, and then they stop. And then if the field is applied again, then the bands move again. So it helps the large piece of DNA to migrate better across the agarose pores. And another one, gradient gel electrophoresis. For example, like this gel is made of acrylamide, okay? That is the material. And usually, usually the acrylamide concentration is homogeneous, same across the gel. But if you apply this technique, then you can give gradient of this acrylamide concentration. Maybe this concentration gets maybe thicker toward the downsides. All right, then in that case, what is the advantage? The advantage of doing that is to resolve fragments that are very close in size. Let's say, like, for example, in normal electrophoresis, you get these two bands very similar in size. Therefore, you will see those bands overlapping, almost overlapping here, so you don't know which one is which. But if you apply this technique, you can a little bit enhance the separation between these two between these two bands. All right. Okay. Now let's enjoy the movie on this agarose gel electrophoresis protocol. It shows the actual experimental method conducted in a lab. So enjoy it.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the die front moves from the negative electrode towards the positive electrode.
So using this electrophoresis technology, we can come up with some very interesting interesting analysis, which is called RFLP, The full name is restriction fragment length polymorphism. This analysis is used for identifying the genetic relatedness between two different organisms. How genetically close between these organisms can be analyzed through this method. So let's say one organism has this type of genetic structure, and there is another organism. It's got this they are very close to each other, right? If you look at the genetic structure, A, B, C, D, E, F, A, B, C, D, E, F, very close. But this number 2 organism is a mutant that has a mutation site here. Number 3. Here, 1, 2, 3, 4, 5. These 5 sites are for restriction enzyme coding sites. So restriction enzyme can code these 5 sites. Whereas these number 2 organism got only 4 sites. The 3 one here, this cutting sites is somehow has been altered and mutated. So the restriction enzyme can no longer recognize this site, therefore cannot cut this site. So if you apply the restriction enzyme, certain restriction enzyme, then we obtain from this enzyme, we get, let me change the color of the pen, hold on a sec. We obtain these 4 different fragments, A, B, C, D, E, F. Whereas from number 2 organism, we can obtain only A, B, C plus D, and E, F. We have 5 different fragments. Here we had 6 fragments. So what do we get after electrophoresis? We get 5 bands. Oh, no, sorry. 6 bands from here, number 1 organism. And from organism number 2, we get how many? How many we get? We should get 5. So 1, 2, 3, 4, 5. So this one here marked as orange color band is a new band from C plus D. Why? Because the largest one should be heavier, the heaviest one. So the heavier one migrates slowly, and the smaller one migrates faster through the electrophoresis gel, as you saw from the animation just before. So the smallest one should be at the bottom, should be positioned at the bottom, and the largest one should be at the top. So from these 2 band patterns, what do you get? What kind of information do you obtain? They look very similar, but one is missing here, and the other one is missing here. So their genetic information is quite similar with some mutation. So if the band pattern is very different from these for these 2 different organisms, that means their genetic information is very different. Therefore, their genetic relatedness is very far. Their genetic information is very different, and they are genetically far away from each other. You understand? Okay. All right. Let's go faster because it's got taking a lot of time. So now we are going to talk about the actual measurement methodology. This one is explaining the method of finding the concentration of DNA. As you already know, the DNA consists of all this nucleic acid, and nucleic acids got some aromatic rings. And these aromatic rings can absorb the light of 260 nanometer. So the higher concentration of DNA you have, then the more absorbance you can see from this experiment. You use UV spectrometry. So if you have higher concentration, of course you should be able to see the higher absorbance. Transmission is the opposite. You shouldn't be confused about that. So if you know the certain concentration of certain sample, you can calibrate beforehand. So you get this profile, and then afterwards, if you have unknown sample of DNA, you don't know, but you got, for example, here, let's say 0.6 observance, then maybe your sample's concentration, DNA concentration, should be somewhere around should be 30, around 30 should be that. So the exact number you have to calculate from the linear equation of this. And from now on, we are gonna briefly discuss each slide of how to label your DNA. Labeling DNA is very important technology because you have to locate certain DNA. For now, you won't be able to understand about that, but just take the knowledge, just take it as it is. Then afterwards, you will be able to understand what this is all about. You have to label the DNA. For example, especially for making probe, but you know, without labeling, you won't be able to see which one is which. So, for example, let's see. For example, if you label the certain DNA here, then you will be able to see this DNA band. Otherwise, you won't be able to see where it is. So labeling is important. Labeling DNA is important in that sense. So how do you label the DNA? There are three methods. Or there are more methods, but the most important ones we are going to discuss here are three different methods. First one is radioactive labeling. What do we mean by label radioactive? Radioactive DNA, labeled DNA uses these two different elements. They are so-called isotope. Usually, they use these. And usually phosphorus is has atomic mass of 31. But the ones we use for labeling it's got atomic mass of 32. What is the difference? This got one more neutron. Therefore, the weight is the mass, atomic mass is heavier than that. So, if you label with this 32 phosphorus, then you can see, you can easily detect this DNA. Another method is labeling with the sulfur. Actually, this place was oxygen, but instead of oxygen, they remove oxygen and then replace the sulfur 35 with oxygen. Usually sulfur is number 32. Sorry, atomic mass of sulfur is 32, but they use 35, heavier one, to label the DNA. So after labeling, the labeled DNA fragments are run on the electrophoresis. So these fragments, you can see, as we observed before, you get lots of different fragments here. And then after finishing that electrophoresis protocol, you place a film on top of the gel, and you keep it in the dark, and the band here embedded in the gel, they move toward the film. And then afterwards, you can the film can be developed and the dark lines here, they appear. They are all radioactive labeled DNA. There are some methods with just radioactive DNA solution. This is another method called scintillation counting method. If you are interested, you can take a look and read the text here. And the second method of labeling DNA is by using a fluorescence tagging. This fluorescent labeling DNA is a simple method. You use this fluorescent tag, just attach them on the DNA, and then once the beam is passing through, then the fluorescence is liberated. You can see this is just general knowledge from the general chemistry, and the energy excites from the ground state, that the electron becomes excited state, and then they go down to the ground state, and the fluorescence is liberated. You can see the color. And the third one, the last one here, is through using biotin. Biotin is actually very cheap method. It's less costly than radioactive or other methods. So biotin is a vitamin from a foxglove plant. You don't need to know all the details about that, but overall, you can synthesize the DNA attached with biotin. Biotin can be manipulated in many different ways. You can attach another enzyme. This whole structure is biotin here, and actually from here, biotin tag the uracil nucleotide. So you can somehow link this protein, and this protein is phosphatase. Phosphatase, what does it do? This digest phosphate. Phosphate is here. This protein breaks this bond eventually. And X, you have to remember, X here in biology, usually, X is the blue dye. When they are linked together, they don't show any color, but once it's broken, once this enzyme breaks this bond, then it is liberated here to emit blue color. So using biotin, you can see here through this whole method, you can see blue color. So so far we saw three different ways. First, radioactive labeling. Second, fluorescence labeling. Third, biotin labeling. You have to remember that. and this is a common knowledge. This is about the terminology. Heating in biology, melting in biology. Melting, what do you mean by melting? Melting is from what? Solid to liquid in general common sense. But here, melting of DNA is to separate this double-stranded DNA into two complementary single-stranded DNA. That is what we call melting when it comes to DNA. So how do you melt the DNA? You heat. Just same thing as melting ice to water. Heat the DNA, then stays this bond. What are these bonds? They are hydrogen bonds, right? These bonds start to break due to the heat. They break from what? Starting from AT bond, because they got only two hydrogen bonds. C and G, they got three hydrogen bonds. They are harder to break. So AT bonds start to break first, and then they are melted like that. And once you cool down again, the complementary strands come together and they bond to become a original double-stranded DNA. So if you have higher GC content, the melting point should be higher. And actually this slide should have come earlier. This is how you transfer the DNA band from the gel, agarose gel, to the membrane or nylon film. This is through the capillary action. You place the filter paper under the gel, and you place some weight. You can stack up some paper towels here, and then you just place some weight. It can be anything to give some weight down to the whole system. And this filter paper wicks the water. It wicks the water. The water goes through toward the center, and then this water gets more moistened, and then the gel goes up due to the capillary action toward the membrane. And that's how you transfer the DNA sample from the gel to the film or some membrane. That's it for the this online lecture for this chapter 3. And in class, you have to give presentation on these topics. These topics will be discussed in the next class, of course, in detail, but you have to prepare the contents of these titles. Purpose and protocol of southern blot, and purpose and protocol of northern blot, and purpose and protocol of western blot, and Jew blot and fish. You have to find the information through the internet or an actual example of application of these technologies. Regarding that, you can find them from YouTube and some other internet sites, and then give some presentation. And I will assign a team for each one of these topics. And thanks for your attention so far, and we will see you in the next class. Have a nice day.